In today's presentation, let us discuss the structural organization of nucleic acids. The double helical structure of DNA, that is deoxyribonucleic acid, was solved by James Watson and Francis Crick in 1953. One important piece of experimental data on which the DNA structure was solved was the X-ray crystallographic pictures obtained by Rosalind Franklin. Another was the observations on DNA base composition made by Erwin Chargaff, now known as Chargaff's rules or laws of base equivalence. Let us now discuss the law of base equivalence, also known as Chargaff's rules. During the late 1940s and early 1950s, the pioneer biologist Erwin Chargaff analyzed DNA from several species and also from different DNA from different cell types in the same organism and found certain types of rules. In particular, he found that DNA in all organisms exhibited an important phenomenon, that is, the amount of purines was found to be equal to that of pyrimidines. In other words, adenine is equal to thymine, that is, the amount of adenine in any DNA sample is equal to the amount of thymine, and that of guanine is equal to cytosine, or A plus G equals C plus D. Though this was done only for DNA by Chargaff, this base equivalence law was found to be true for double-stranded RNA later on. However, the true understanding of Chargaff's rules came only after the elucidation of DNA structure by J.D. Watson and F.H. Crick in 1953. Levels of nucleic acid structure. Like proteins, nucleic acids may also have 4 levels of structural organization: primary, secondary, tertiary, and quaternary structures. Thus, for DNA, we may refer to primary, secondary, tertiary, and quaternary structures of DNA. Similarly, RNA molecules may assume primary, secondary, tertiary, and quaternary structures. Primary structure of DNA. Primary structure refers to the linear sequence of nucleotides that are linked together by phosphodiester bonds. DNA is a biopolymer made of units called nucleotides. A nucleotide consists of a base, a sugar, and phosphate. The nitrogenous base is derived from either purine or pyrimidine. In DNA, the sugar is deoxyribose. The phosphate group is derived from phosphoric acid. The primary sequence may be represented by shorthand notations such as A, C, G, T, A, or a schematic diagram as shown in the accompanying figure. The sequence is usually written in the 5' to 3' direction. A DNA chain consists of alternating units of deoxyribose and phosphate. The 3' hydroxyl group of one sugar is joined to the 5' hydroxyl group of the next sugar by a phosphodiester bond. The phosphodiester backbone lies on the outside and the bases are placed inside, stacked on top of each other. Next, secondary structure of DNA. Secondary structure is the set of interactions between bases, that is, which parts of strands are bound to each other. In DNA double helix, the 2 strands of DNA are held together by hydrogen bonds. The nucleotides on one strand base pairs with the nucleotide on the other strand. The 2 strands of DNA are complementary, intertwined, and antiparallel. This means that if A is there on one DNA chain, there will be T on the other chain and vice versa in the corresponding position. Similarly, C will pair with a complementary G residue and vice versa. The 2 chains coil around each other and they are antiparallel, that is, the phosphodiester backbone of one chain runs in the 5' to 3' direction, whereas in the other chain, it runs in the 3' to 5' direction. Now, let us discuss the polymorphic forms of DNA. DNA's secondary structures include 3 polymorphs, A and Z DNA. B is the predominant form in cells. A is usually thicker and Z is thinner and have zigzag backbone. B DNA is right-handed, has 10 bases per turn and has a diameter of 20 angstrom, whereas A form is also right-handed with 11 bases per turn and a diameter of 23 angstrom, and Z DNA is left-handed and has 12 bases per turn with a diameter of about 17 angstrom. There are also other polymorphs such as cDNA and other forms of DNA. An important force stabilizing DNA structure, as has been explained earlier, is the hydrogen bonding between complementary bases. AT base pair consists of 2 hydrogen bonds and GC base pair has 3 hydrogen bonds between them. The van der Waals interactions of the stack bases contribute to the stability, and also the electrostatic interactions between negative charges on the phosphodiester backbone and the positively charged counter ions such as magnesium ion. Triplex and quadruplex DNA. DNA can sometimes also exist in 3-stranded form known as triplex DNA and 4-stranded form known as tetraplex DNA. Triple-stranded or triplex DNA is one in which 3 oligonucleotide chains coil around one another and form a triple helix. In this unusual structure, one strand binds to a B-form DNA by Hookstin hydrogen bonds. In quadruplex DNA, also known sometimes as Z-quadruplex, 4 DNA strands interact to form a tetraplex. Such quadruplex structures usually occur in DNA chains having high proportion of guanine residues. The orientation of the strands in the tetraplex may vary between various types of parallel and antiparallel arrangements. Next, tertiary structure of DNA. Tertiary structure refers to the locations of the atoms in 3-dimensional space, taking into consideration geometrical and steric constraints. It is a higher order structure than the secondary structure, in which large-scale folding in a linear polymer occurs and the entire chain is folded into a specific 3-dimensional shape. There are 4 areas in which the structural forms of DNA can differ. Number 1, handedness, whether the chain is right-handed or left-handed. Number 2, length of the helix per turn. Number 3, number of base pairs per turn. And number 4, difference in size between the major and minor groups. The common tertiary structures of DNA are supercoiled DNA and various forms of chromatin structure. Supercoiling results from change in the so-called linking number of DNA. Linking number, which is represented by the symbol L, is a sum of twists T and Rit, also known as Riting number W, that is L equals T plus W. Twists are the number of times the 2 strands of DNA are twisted around each other. Rits are the number of times the DNA helix crosses over itself. DNA in cells is negatively supercoiled and has the tendency to unwind. Hence, the separation of strands is easier in negatively supercoiled DNA than in relaxed DNA. The 2 components of supercoiled DNA are solenoid and plectonemic. The plectonemic supercoil is usually found in prokaryotes, while the solenoid type of supercoiling is mostly seen in eukaryotes. Next, quaternary structure of DNA. The quaternary structure of nucleic acids is similar to that of protein quaternary structure. Although some of the concepts are not exactly the same, the quaternary structure refers to a higher level of organization of nucleic acids. Moreover, it refers to interactions of the nucleic acids with other molecules such as proteins. The most commonly seen form of higher level organization of nucleic acids is seen in the form of chromatin, which leads to its interactions with the small proteins called histones. Also, the quaternary structure refers to the interaction between separate RNA units in the ribosome or spliceosome. Primary structure of RNA. The sequence of nucleotides in the RNA chain. RNA differs from DNA in having uracil in place of thymine and ribose sugar instead of deoxyribose in DNA. Next, secondary structure of RNA. RNA usually exists in single-stranded form but can sometimes assume double-stranded forms. When single-stranded, RNA does not obey Chargaff's rule, but double-stranded RNA obey Chargaff's rule just like DNA. That is, the proportion of A will be equal to that of U and that of C will be equal to that of G. In other words, A equals U, G equals C, A plus G equals C plus U. RNA-DNA hybrid structure is similar to that of aDNA. Secondary structures of RNA molecules include hairpins, bulges and internal loops, etc. A bulge will form in a RNA double helix when there are non-complementary bases on one strand. If there are non-complementary bases on both strands, it would lead to formation of internal loops. A stem loop, that is hairpin structure, will form when the two RNA chains have complementary sequences forming the stem of the hairpin with non-complementary bases forming the loop. tRNA. tRNA usually has 4 double helical regions. Phenylalanine tRNA assumes L shape. There are many unusual bases present in tRNAs. Ribosomal RNA, rRNA. In rRNA, there is high intra-strand sequence complementarity leading to extensive base pairing. Tertiary structure of RNA. The overall shape of the RNA molecule. One common example is that of the phenylalanine tRNA. It shows L shape in the tertiary structure. Similarly, hammerhead ribozyme, that is an RNA molecule with catalytic function just like enzymes, displays unique three-dimensional or tertiary structure that determines its function. In this manner, ribosomes, which are protein synthesizing machineries, and spliceosomes, which are involved in RNA processing, also have specific tertiary structures that influence their functions of protein synthesis and RNA processing respectively. Next, quaternary structure of RNA. The quaternary structure of RNA is displayed by higher order entities formed by association of RNA with proteins. For example, many viruses have unique quaternary structures in which proteins form the viral capsid cell that houses the RNA molecules that form the genome of the virus. Structural organization and nucleic acid functions. DNA structure and function. Structure and function is intimately related in biology. DNA structure influences its function. For example, the usual genetic function of DNA is determined by the structure of bDNA, which predominates in the cell. RNA structure and function. The same structure-activity relationship applies to RNA too. tRNA structure influences its function of carrying amino acids to the site of protein synthesis. Similarly, the unique structure of ribozyme or ribosome influences its catalytic or protein synthesis functions. Conclusion. In today's presentation, we have had a detailed look at the structural organization of nucleic acids. We have seen that both DNA and RNA may display 4 levels of structure. Primary structure is the sequence of bases in the polynucleotide chain. Secondary structure is the conformation of the polynucleotide backbone, whereas tertiary structure is the overall three-dimensional shape of the nucleic acid molecule. Quaternary structure is the highest level and is usually displayed by DNA or RNA in association with other molecules such as proteins. We hope that you had a conceptual understanding of the levels of nucleic acid structure, and we wish you success in your further academic odyssey. Happy learning. Thank you very much.